Today we'll find out about protein denaturation using this guy by doing 4 experiments. But first, let's check our proper protective equipment.
Lab gown, gloves, tie your hair, wear your hairnet. Wear your mask properly. Lab goggles. Now that we're all set, let's go! First and foremost, separate the egg whites from the yolk. Measure 15 ml of egg whites. We'll be using this for all the experiments. The first experiment involves mechanical agitation. We'll be using bowl, syringe, fork, and egg whites. Measure 5 ml of egg white and put it in the bowl. Whisk it vigorously with the fork until white. Whipping egg whites incorporates air, causing the proteins to stretch and create bubbles. As compared to the normal one, this one has more volume, is stiffer, and more opaque. The second experiment involves heat. We'll be using oiled frying pan, egg whites, syringe, spatula, and plate. Measure 5 ml of egg white and cook it in the pan at very low heat for a minute. Transfer to a plate once done. Heat forces the liquid state to turn solid due to collision and reorganization of proteins with water molecules. As compared to the normal one, this one is more opaque, solid, and rubbery since it's been cooked. For experiments 3 and 4, we'll be using the egg albumin solution. Mix 5 ml of egg white in 45 ml of water. The third experiment involves alcohol. We'll be using medicine cups, syringe, 70% alcohol, albumin solution, and fork. Measure 2 ml of albumin solution and put it in a container. Add in 5 ml of alcohol. Stir and leave for 5 minutes. Alcohol disrupts the protein bonds and form new linkages with each other. As compared to the normal ones, this one formed some wispy opaque whites. The last experiment involves acid. We'll be using medicine cups, syringe, vinegar, albumin solution, and fork. Measure 2 ml of albumin solution and put it in a container. Add in 5 ml of vinegar. Mix thoroughly and leave for 5 minutes. Adding acid deforms the protein and form bonds with other proteins, causing them to coagulate. As compared to the normal one, this one formed feathery strands of whites.
And we're done! Now, for a few takeaways. Egg whites are high in tightly bound proteins such as albumin, mucoprotein, and globulin. Whenever an external stressor is applied, in our case, agitation, heat, alcohol, and acid, protein structure in the egg whites begins to unravel. When these bonds are disrupted, its natural function will be altered. This whole process itself is called the protein denaturation. Now go have fun and boast your new knowledge to everyone. See you around!